Initial formation of synaptic connections. Acquiring areal and laminar positions and phenotypes comprises only the first and most fundamental steps in the formation of species-specific cortical development. However, an equally important next step is the formation of synaptic connections. It is a very large subject, is studied by most developmental neurobiologists, and by itself deserves more than a short section. It is in part covered in the subsequent chapters, and it is only briefly reviewed here to emphasize that in primates, and particularly in the human, it is a prolonged process that involves overproduction of cells, axons, and synapses, and their later elimination in response to environmental influences. This process is often called neuroplasticity, and we consider it as a phase of the normal course of brain development. In the macaque monkey, it peaks after birth and lasts at least 3 to 4 years, and in the human, it lasts 15 to 19 years. Detailed studies in the rhesus monkey showed that both neurons and their axons are overproduced in the cerebral cortex during well-delineated stages of development. For example, there are about 40% more neurons in the monkey visual cortex during the second half of pregnancy than in the adult. Furthermore, a newborn monkey has almost 200 million callosal axons compared to less than 50 million in the adult. The axons are lost at a rate of about 8 million per day or 50 per second during the first 3 weeks after birth. Thereafter, they are lost at an estimated rate of 500,000 per day or 5 per second until the adult value is reached. Similar magnitude elimination of callosal axons has been reported for other mammalian species. The functional significance of this loss of axons is not fully understood, but the prevailing hypothesis has been that activity-dependent stabilization plays a critical role. In the rhesus monkey, during the first 2 to 3 months of postnatal life, synaptic density increases rapidly and reaches a peak that is about 2 times higher than in the adult. This synaptic density, well above the adult level, lasts through infancy and adolescence. We have calculated that about 1.8 times 10 to the 11 synapses are lost in the visual cortex of a single cerebral hemisphere during puberty in the monkey. The magnitude of this decline is stunning, when expressed as loss of about 2,500 synapses per second during this period. Since other areas, including association cortices, simultaneously undergo comparable synaptic loss, more than 30,000 synapses per second are deleted from the entire cortical mantle during monkey adolescence. In the human, in whom this period of life lasts about 3 times longer, but the cortex is 10 times larger, the synapses may be lost at a rate of 100,000 per second. The decline in synaptic density is due primarily to elimination of excitatory junctions located on dendritic spines, while inhibitory synapses on dendritic shafts remain relatively constant. Other results showed that the density of major neurotransmitter receptors in the cortex also reaches a maximum level between 2 and 4 months of age, and then declines to the adult level during the period of sexual maturation. These findings revealed an unusual coordination between biochemical and structural differentiation and indicated that these events may be related to maturation of function. The prolonged phase of postnatal development in primates provides unparalleled opportunity for competitive, activity-driven stabilization among initially supernumerary inter- and intracortical connections. The formation of the final pattern of cortical connections is achieved through dynamic interactions that involve at least 2 well-defined steps. The first step, one set of axons project to the target structure guided by pre-specified short-lived molecular markers without regard to specific location on the individual neurons or their parts. In the second step, which is activity-dependent, synaptic connections are sorted out and remain on only selective sets of neurons or their dendrites. Our experimental studies of the formation of binocular vision in primates provide a dramatic example of this biphasic development and support the hypothesis that competitive interactions between 2 or more populations of neurons play a significant role in the elimination of axons and segregation of their synapses. This phase of cortical development has a profound significance for understanding development of human cognitive abilities. Some implications are obvious. For example, Our educational system, which postpones disciplined intellectual learning until later stages in life, may not be biologically optimal, as the first 15 years may be the most important formative phase as far as synaptic stabilization is concerned. It is well known that high-level professional musicians or athletes can be created only at a specific critical age, and there is no reason why other cognitive skills essential for human intellectual abilities are not set up also at this time. Our data provide biological support for intensive targeted intellectual training that starts a child